Good morning, students. This is Murthy sir. In the last class, we discussed about the functions of plasma membrane, diffusion and osmosis concepts, practical examples, Graham's law of diffusion, reverse osmosis, its practical application. Today, we will learn about some practical observations related to osmosis. Students, osmosis can be defined as the movement of solvent from less concentrated solution to high concentrated solution by crossing semi-permeable membrane. The aim of the experiment is to observe endosmosis and exosmosis in raisins or dried grapes. The materials required are 2 beakers, tap water, sugar, dry grapes or raisins. Students, in this picture, you can observe a raisin in a beaker containing water. Students, when water enters into the raisin, it is called endosmosis. When water leaves the raisin, then it is called exosmosis. Now let us see the procedure. Take a raisin and drop it in a beaker containing tap water. Leave it for 1 hour. Now let us see the observation. You will observe a change in the size of the raisin. The raisin will swell due to endosmosis. Water enters into the raisin. Students, generally all the fruits are sweet in taste due to the presence of a sugar called fructose. It's an organic compound. Students, in this experiment, water is entering into the raisin from the beaker by crossing cell membrane due to the presence of some sugars in it, that is fructose. In this table, you can observe fruit sugars analysis. Apple, banana, grapes, orange, carrot, bell pepper, sweet potato, table sugar. Students, in this table, the percentage of sugars in grams it is mentioned. Free fructose, free glucose, and total amount of sucrose in grams it is clearly mentioned. Students, now let us see the conclusion of the experiment. The raisin being a fruit, it contains fructose sugar, so solvent moves through the cell membrane and enters into the raisin. That is, solvent moves from less concentrated region to more concentrated region. So as a result, endosmosis happens. Students, now in the second stage of the experiment, when you leave this swollen raisin in a beaker containing saturated sugar solution overnight, then the swollen raisin gradually shrinks. Here exosmosis happens. The swollen raisin, when we keep in a beaker containing saturated sugar solution and leave it overnight, then exosmosis happens. The raisin shrinks due to movement of water from raisin into the beaker containing sugar solution. Students, you know what is a saturated sugar solution? When the amount of solute that is dissolved in water reaches to the maximum level, then it no longer dissolves in that water, and that is called saturated sugar solution. Students, now let us move on to the next experiment, potato cup experiment. The aim of the experiment is to observe the process of osmosis. The materials required are 1 raw potato, 1 boiled potato, 2 beakers or bowls, 2 pins, water, sharp knife to cut the potato. Procedure. Take a raw potato and a boiled potato. Peel off the skins, cut cube-shaped cups from them as shown in the figure already. Students, here we have to make a potato cup to keep sugar solution in that. And in the petri dish, we have to take water, tap water. And we have to observe how the process of osmosis happens here. Students, this is the potato cup made, and in this we had to take sugar solution, and this we had to keep in a petri dish containing water. Students, now let us see the procedure. Prepare a few amount of saturated sugar solution. Pour the sugar solution in the potato cup to a level as shown in the figure already. Mark the level of sugar solution by piercing the pen. Keep the potato cup in the petri dish containing water. Students, this arrangement we have to leave for half an hour, and then we have to observe what happens in this experiment. Students, in this experiment, water moves from petri dish into the potato cup due to osmosis. The petri dish contains less concentrated solution, and in potato cup, sugar solution is there which is more concentrated. So solvent moves from less concentrated solution to more concentrated solution by crossing the cell membrane. So this is osmosis. Pour water in the beaker or bowl to half of the height of the potato cup. Ensure that the potato cup does not float or submerge in water. Leave the arrangement for half an hour and note down your observations. Students, in this experiment, solvent moves from beaker into the potato cup. The reason is the water that is present in beaker does not contain any solute particles in it. So the water molecules are in a free state, so they are able to move freely into the potato cup. Whereas the potato cup contains sugar solution. There sugar is present as solute. So along with water molecules, sugar molecules are present there. So it is a concentrated solution.